You're listening to Medscape's In Discussion series on endometrial cancer, a podcast where thought leaders and clinical experts share their diverse insights and practical ideas for optimizing patient care. This series is hosted by Dr. Ursula Matulonis, Chief of the Division of Gynecologic Oncology at the Dana-Farber Cancer Institute and Professor of Medicine at Harvard Medical School. She is the first recipient of the Brock Wilson Family Chair at the Dana-Farber Cancer Institute. She co-leads the Gynecologic Cancer Program within the Dana-Farber Harvard Cancer Center, the Ovarian Cancer Specialized Program in Research Excellence, SPORE grant, from the National Cancer Institute within the Dana-Farber Harvard Cancer Center, and an NCI P01 Program Project grant that is focused on targeting replication stress in high-grade endometrial cancers. Relevant disclosures can be found with the episode show notes on Medscape.com or the Medscape app. The topics and discussions are planned, produced, and reviewed independently of advertisers. This podcast is intended only for U.S. healthcare professionals.
Hello, I'm Dr. Ursula Matulonis. Welcome to Season 2 of the Medscape In Discussion Endometrial Cancer podcast series. Today, we'll discuss antibody-drug conjugates and how they fit into gynecologic cancers, but also importantly, hearing from our guest, Dr. Leif Ellisen, about different aspects of ADCs that perhaps we haven't been thinking about in GYN cancer, or we're going to have to think about really closely soon. It really is an honor to have Dr. Ellisen here today. He's a professor of medicine at Harvard Medical School and program director for breast medical oncology. He's also the clinical director of breast and ovarian cancer genetics at the Mass General Cancer Center. He's also the co-leader of the breast cancer program at the Dana-Farber Harvard Cancer Center. Welcome to the Medscape In Discussion Endometrial Cancer podcast.
Dr. Matulonis, Ursula, it's great to be with you and looking forward to discussing this important and fascinating topic.
So, as you know, there has been an explosion of different antibody-drug conjugates and oncology in general, and these drugs are now making a significant impact in GYN cancers with FDA approvals for mirvetuximab in ovarian cancer, the tumor-agnostic accelerated approval for trastuzumab deruxtecan for HER2 3+ advanced cancers, which really applies to all GYN cancers, and then tisotumab for advanced cervical cancer. There are also multiple phase 3 trials, as well as earlier phase 1 and 2 trials of antibody-drug conjugates in gynecologic cancers, but in endometrial cancer specifically. These antibody-drug conjugates are targeting several different payloads, such as HER2, TROP2, folate receptor alpha, CDH6, and some have the payload of topoisomerase 1, whereas some have antimitotic payloads. We're already seeing in phase 1 trials of topo-1 payloads that previous exposure to previous topo-1 payloads are not allowed, and we are eventually going to have to think about rationally how to sequence different antibody-drug conjugates and really understand the mechanisms of resistance and response to each of these drugs that we're going to be using. Leif, I really want to start and just really thank you for being here today. You and your team at Mass General are leaders in antibody-drug conjugates, and you had a paper recently in Clinical Cancer Research identifying the emergence of topoisomerase 1 mutations as a resistance mechanism to topo-1 payload antibody-drug conjugates. So I really want to focus on resistance mechanisms and ask you, what do you think are the most common and impactful resistance mechanisms that cancers develop to antibody-drug conjugates, and some of the challenges to using ADCs, not just once, but repetitively?
Well, thank you again, Ursula. These are very important questions. And I think one thing to set the stage for the discussion is that ADCs are quite complex molecules. When you think about an antibody binding on the cell surface, you think about delivering some kind of payload that's bioactive, and you think about how the antibody is linked to the payload, which determines how it's delivered. So there are multiple and complex resistance mechanisms that develop. One way to divide them up would be de novo resistance versus acquired resistance. Some of the work that we're doing in de novo resistance suggests that you can see really a lot of intrinsic chemoresistance of the tumor cells, just like you would with standard chemotherapy as a mechanism. In some cases, low expression of the ADC target itself, although that doesn't seem to be a major contributor. More interestingly, we're learning that in de novo resistance, many features of the tumor microenvironment that may affect both the biology of the tumor cells and the penetration of the ADCs into the tumor are important and may confer resistance. Now, when we look at the acquired resistance setting, surprisingly, we don't see a lot of loss of the target per se, although we and others have demonstrated where loss or mutation of the target likely mediated acquired resistance. More commonly, we see some of the things that you might see with chemotherapy, such as activation of cell survival pathways in the tumor, upregulation of drug efflux transporters, and mechanisms of that kind. More interestingly, though, and perhaps specific to the ADCs, is specific mutations of the target of the payload. For example, as you mentioned, topoisomerase 1, which when administering systemic topo-1 inhibitors, you don't really see emergence of these mutations, but we are seeing them now in the setting of the ADCs, which may have to do with the fact that we're really just delivering much higher doses, it's more potent, and the tumor is pressured to have an on-target resistance mechanism.
That's very interesting. And I think that as we sort of step back as clinicians to try to navigate this, in, as you mentioned, both the de novo and the acquired mechanisms of resistance, what are some of the strategies that you're pursuing, both from a laboratory standpoint, but also from a practical clinical standpoint, how to overcome these ADC resistance mechanisms?
My bias since we started working on ADCs a number of years ago was that because of the complexity of these molecules, that a lot of the work that we needed to do was going to be analysis of patient samples themselves to really understand, among all the possible ways that resistance could occur, which are the ones that really occur importantly in vivo. And so, for example, we've launched a number of preoperative trials. This is in breast cancer, where this is a common method for treatment now, where we can profile the tumor before treatment and after treatment. We can identify through things like single cell analyses these tumor phenotypes and tumor states that are chemoresistant. Then we can do things like spatial analysis to identify what we call cellular neighborhoods or ecosystems within the tumor that governs how the tumor cells themselves behave. Are they hypoxic and chemoresistant? But also whether the drug can make its way into the tumor, governed by things like angiogenesis, which may preclude effective delivery of the ADC. And then when we switch to thinking about how we can study mechanisms of acquired resistance, one very useful tool has been the increasing use of circulating tumor DNA analysis, that is blood-based analysis, because we can do repetitive sampling in the setting of progression and get really an overview of the genetic evolution that gives rise to resistance. And that, for example, was how we identified these topoisomerase 1 mutations. But it's also the case that we need to look at tissue. That can be challenging in the metastatic setting. And so what we've done there is develop a number of patient-derived resistant tumor models that we can then take back to the lab, propagate them, understand resistance mechanisms, and then do things like really complex and comprehensive screens to ask in an unbiased way, how might that resistant state be overcome? And I really think fundamentally it's going to take this kind of coordination between the laboratory and the translational work that's going to give us the best insights that are going to be most relevant to patients.
Before we move on to biomarkers, I just wanted to ask you the question, given that you've identified these topoisomerase 1 mutations and changes, because this is important, because this is a very commonly used payload, of how to overcome, or do you think it's possible to overcome these different resistance mechanisms clinically?
Well, I think topo-1 is a particularly fascinating one because, as we know, topoisomerase 1 itself is so essential to fundamental processes in the cell, transcription, replication. And what happens with a topo-1 mutation is the enzymatic activity of topo-1, it's not lost, but it's substantially modified. And that modification of the activity requires the cell to adapt in very complex ways. And we're just beginning to understand it. But we do believe that that adaptation that results from, for example, a topo-1 mutation is going to create additional vulnerabilities in that tumor cell. And so we're doing a lot of work, and I know a lot of others are as well, to try to understand those evoked vulnerabilities that occur after such resistance mutations that might lead to ways to overcome that particular state.
That's really fascinating. So I'm going to move on to biomarkers. And, what we're seeing in GYN, and I don't think we're as far along as in some other cancers, so we have a lot of work to do, but really understanding the heterogeneity of the biomarker expression in different patients, but also different sites of recurrence. And this really happens in endometrial cancer where patients will have sometimes, peritoneal disease resulting in ascites. They may have lung metastases. They may have nodal metastases. And sort of how are you thinking about the biomarker changing over time within a patient and within their different metastatic sites?
Yes, of course, this is the big challenge of advanced and metastatic cancer as a whole. Just to give you an idea of what we're facing, a number of years ago, we published a paper describing that in an individual breast cancer patient had received a sacituzumab govitecan. They had a really fantastic response to the drug, but progressed at multiple sites. And we were able to do individual sampling of those sites. And in one subset of metastatic sites, there was a mutation in TROP2, which was the target of the ADC, which caused loss of expression and resistance to those particular sites. And in another subset of metastases, mutation of topoisomerase 1, again, resistance to the payload. We hope that this kind of phenomenon, will not be occurring in all of the patients. And I do believe that to the extent that we can make ADC therapy more effective and more potent, that we have a better ability to combat this heterogeneity that's intrinsic to the cancer. And that might be through, better selection of ADCs. That might be through combinations that we might talk about momentarily to really try to expand the way that the body responds, expand the ability to target the tumor, in particular through combinations such as immunotherapy, where you can then activate the immune system to combat the heterogeneity of the metastatic tumor. But there's no doubt that it's a daunting challenge for the whole field.
Yeah. And I just wonder if, in terms of how we use ADCs in a patient's treatment and where that patient is at the start of their cancer journey versus later on, obviously likely resistance mechanisms as the tumor changes over time become increased. So one would think that, and this happens clinically, right, where the ADC that you use first probably has the best chance of response. And should we be thinking strategically about how to position the ADC sequencing?
Yes, that's exactly right. And there's no doubt that we have to be smarter about that. For example, coming back to these topoisomerase 1 mutations, it seemed to be quite clear from the relatively limited but quite compelling clinical data so far that if you have one of these mutations, you're very unlikely to respond to a second ADC that harbors a topoisomerase 1 inhibitor payload. And so I think that's a general lesson that the more we can understand resistance, the more we can test for resistance at the time of progression on one ADC, the more likely we are to be able to make a better choice in the second ADC. And as we can imagine, because ADCs, as you mentioned, are being tested in so many different settings, and there's going to be numerous ADC options in the future, we'll have many ADCs to choose from, and making the smart choice, hopefully, will be informed by knowledge about specific mechanisms that arise in individual patients.
Yeah, that totally makes sense. So what do you see as the differences and differentiators regarding the other parts of the antibody drug conjugate? And I know you mentioned this a little bit before, but specifically the linker and then the type of the antibody.
Yeah, well, these are really important considerations and speak to the complexity of these ADCs. So the linker, again, linking the antibody to the payload, is important because it determines how much of the payload is released outside the tumor cell. So if you take two examples on the extremes, sacituzumab govitecan has a pH labile linker, so it's quite labile, short half-life, and so you have a lot of payload, SN38, a topo 1 inhibitor, released into the circulation, and therefore sacituzumab has a lot of chemotherapy-like toxicities. On the other end of the spectrum, you have an ADC like trastuzumab emtansine, TDM1, which has quite a stable linker that requires intracellular processing and lysosomal processing to release the payload. And so there, you can see how a very labile versus very stable linker controls the side effect profile to a large degree of these two drugs. It turns out features of the antibody are also quite important. And one of the particular ones that's becoming into focus is the issue of once the antibody binds, whether it stays on the cell surface or whether it's rapidly internalized. Again, going with SG, you have rapid internalization, and so no ability for the antibodies to stay on the surface. And we know that antibodies that coat the surface of tumor cells attract immune molecules that can induce things like antibody-dependent cellular cytotoxicity and phagocytosis that can engulf and kill tumor cells. So you have this balance between rapid internalization, which can deliver more payload, and the staying on the cell surface, which can recruit the immune system. And so There's not a perfect formula or secret sauce for what everybody believes is the best way to design your antibody. And these are things that are being tested systematically to see whether there really is a best practice or best design along those lines.
Yeah, it's really interesting. A lot to learn about all these different ADCs and really how unique they are. And you really just never know until you start testing them on patients and seeing, hopefully they're safe and effective. This is really exemplified in breast cancer, but there are some ADCs, and hopefully we'll see this in GYN cancers as well, but there's some ADCs that really don't require minimum levels of biomarkers, such as we've seen with HER2 and trastuzumab deruxtecan, TROP2, as you've mentioned, CD6, and in certain circumstances, FOLR1 as examples. And so is the efficacy of these antibody conjugates really about the presence of just maybe tiny low levels, despite there being maybe 0 or 1 plus? Or is there some other mechanism of action that these ADCs are imparting against the cancer cell? These are questions really around where the field is headed, because clearly, not requiring tissue, not requiring a biomarker is much easier for clinicians and for patients than requiring perhaps new biopsies, sending testing out for biomarker assessment, et cetera. So just wondering what your thoughts are about that.
Yeah, so again, this is a really fascinating aspect of ADCs. And currently, there's a sort of debate in the field of ADCs about, in general, how much of the ADC activity is really related to, this canonical ability to bind to the target and deliver into the tumor cell, and how much of ADC activity relates to a sort of a slow and persistent payload release in the microenvironment, kind of what we might call a metronomic release of payload in the tumor microenvironment that is going to be less target dependent, or particularly less dependent on very high levels of target. I would say that undoubtedly most ADCs exhibit some aspect of both of these, and you see this a little bit in clinical data where at very high levels you have a correlation with target expression and duration of response, but at lower levels there's not a great correlation. So both of those exist depending on the design of the ADC and the target itself. And which is better, high specificity and target dependence versus not screening? I would say that in the short term, it's certainly convenient, as you mentioned, that we have ADCs that don't require pre-screening. But I do think in the long term, particularly as we have more ADCs becoming available, it would be quite valuable to identify patients who are highly likely to benefit from a given ADC based on target expression. So in other words, as more ADCs become available, it would be valuable to have a means to select the best ADC among many for a given patient, either by the target expression or some other biomarker, the personalized patient-selected cancer therapy that we all like to give. Ultimately, with a lot of ADCs available, I think that's what we'd like to do.
As the ADC is sort of coming into near the cancer cell, that what happens, either that internalization or perhaps attraction of other molecules or other cells to that interaction, really would depend upon perhaps the target. So maybe B7H4, which may have a more immune response, perhaps, versus also where the cancer is. Is it located nodally, or is it somewhere else where maybe there are not as many immune cells, perhaps? Or do the immune cells just come regardless of where the cancer cell is? Any thoughts about that?
I think it's going to be important, and I agree with you. And I think it's going to be particularly important as we think about some of these more novel combinations, for example, payloads that may activate the immune system, combinations with immune checkpoint inhibitors. And I think their site-specific tumor immune microenvironment and context are going to be even more important drivers of the responses that we see.
Yeah. And I think that was another one of my questions to you, is really around what types of combinations of either ADC or ADCs plus something else are you most excited about?
Yeah, I think it's a very exciting time because I think we have so many opportunities to combine ADCs with, as you said, with other ADCs potentially, but with other molecules and therapies and even established therapies that are mechanism-based and potentially synergistic, and even potentially combinations which might have been tried in the past but didn't work because of prohibitive toxicity. One example is a clinical trial that we did with sacituzumab govitecan together with a PARP inhibitor. It was known that topoisomerase 1 inhibitors and PARP inhibitors were synergistic systemically, but the problem was there wasn't a great therapeutic window because it was highly toxic to hematopoietic and other cells. But we reasoned that the specific delivery to the tumor cells might give more specificity and widen the therapeutic window. Turned out that was only partially true. And in fact, we had to do the sequencing of the true drugs, the sacituzumab and the PARP inhibitor, given sequentially, not at the same time, but still the idea that this kind of combination, which if you deliver just systemic drugs, totally prohibitive, could be feasible. And we were able to complete the clinical trial successfully with an ADC-based delivery. And then, as I mentioned earlier, I do think that in the near term, there are already many clinical trials going on combining ADCs with immune checkpoint blockade. I think this is particularly exciting because we know that one of the major features that can stimulate the immune microenvironment is immunogenic cell death. And we do know that ADCs are really effective in many cases at inducing this kind of cell death and potent tumor responses, which is a great stimulation to the immune system. And then combining with checkpoint inhibitors in the right context, I think is a very exciting possibility, at least in context. And we're going to be having more data emerging about that in the coming months and years.
Yeah, that's really exciting. That's great. I want to move to ADC payloads and really focus on two questions here. Obviously, the ADCs that we're using mostly clinically are either comprised of topo-1 inhibitors or antimitotic agents. So what do you think are some of the future payloads that are unique and different from these two? And then also, much is discussed about drug antibody ratios or DARs. And everyone thinks higher is going to be better, but sometimes higher is toxic. And what are your thoughts about the sweet spots of the DAR, or does that not matter as much?
Starting with payloads, I think we shouldn't overlook the ability to just try new chemotherapy payloads. I think one of the reasons I believe that in breast cancer, the proved ADCs have been so effective, these are all bearing topo-1 inhibitors by and large, is because we've never used topo-1 isomerase inhibitors in breast cancer. So typically no breast cancer has ever seen that drug before. And then we come in with an ADC and a totally new chemotherapy that can be successful. So leveraging on that paradigm and linking the ADCs to other chemotherapies, which we know can be successful in particular contexts, is certainly one thing that's being tried and that's reasonable. Others, I think, very exciting things like immunomodulatory agents and immune agonists, for example, delivering those in a specific way. We know that systemically, immune system agonists have been tried, but in some cases have prohibitive toxicity, but when delivered selectively might be quite successful. So conceptually, very interesting. And then other classes of target inhibitors that we know have efficacy in the right setting, but again, might have been shown to be relatively toxic systemically, such as various DNA repair inhibitors, DNA damage checkpoint inhibitors. All these things, given our ability to deliver them in a more specific way with ADCs, I think now can be considered and reconsidered. And, that's a very exciting potential, I'd say.
It's really quite impressive to think about this field moving forward.
I will do a comment on the DAR because I think that's a drug to antibody ratio, because I think that's another fascinating one. And I think the field has not settled on an answer, because when you think about it, if you increase the drug to antibody ratio, what it does is mean that you're actually infusing fewer molecules of antibody, right? Because if you have more drug per antibody, you're infusing fewer molecules of antibody for a given toxicity that can be tolerated. And what that means is that there's a consideration called the saturation front, where if you have a lot of target in a certain part of the tumor, if you don't have enough antibody molecules there, they get saturated. And so you never get the tumor penetration to actually kill all of the tumor, and that leads to generation of resistance. On the other hand, lower DAR, more antibody, you can overcome that saturation front, you can get into the tumor, but maybe not as potent per ADC molecule. So there is a sweet spot there, whether it's going to depend on the individual payload and target or the individual tumor type, I think remains to be seen, but there's going to be a lot of investigation of that area, and it's going to be a really fascinating one to watch.
I want to finish up by asking you to tell us about some of the ADC research that you're working on right now that you're really excited about.
I think the real excitement in ADCs comes from their complexity, and it's the opportunity to really merge what we're seeing in patients about how response and resistance are governed with what we can learn in a systematic way in the laboratory. When we think about de novo resistance, neoadjuvant studies, understanding evolution and microenvironmental determinants that we typically don't think of when we're thinking of drug resistance. We think of the tumor cell, but thinking about how the individual cells are arranged into a neighborhood within the cell, maybe not top of our list for determining whether we're going to give a drug or not. But I think it may be in the setting of ADCs. And then secondly, this idea that we can really think about mechanistic and rational combinations with ADCs that might not have been possible before for toxicities or other reasons, and then allowing the selective delivery that we can get with ADCs to make this possible and really get potent and synergistic tumor cell killing in the way that we always hope to do.
Really, what an exciting field. And, congratulations to you, Leif, for all you've done, your team has done, and you're really pioneers in this field. So I think we're, once again, GYN cancers are learning from breast cancer, and you're leading the way on this. And really, thank you so much for being here today. It's a pleasure to see you. It's great to chat with you. You're such a great colleague and friend. So today we've talked to Dr. Ellisen about ADC resistance mechanisms, ADC sequencing a little bit, talking about the different components of the antibody-drug conjugate, what's exciting in the field, novel payloads. Thank you so much for tuning in. Please take a moment to download the Medscape app to listen and subscribe to this podcast series on endometrial cancer. This is Dr. Ursula Matulonis for the Medscape In Discussion Endometrial Cancer Podcast.
Thanks for listening to Medscape's In Discussion Endometrial Cancer podcast series with our host, Dr. Ursula Matulonis. Be sure to look for more In Discussion episodes wherever you get your podcasts. Check out Medscape.com or the Medscape app for show notes, links, and more information on endometrial cancer.